Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, let's unpack something that might be hidden in plain sight in your kitchen. Ever reach for a low-sugar protein bar? Maybe a diet drink or a treat marketed for managing blood sugar.
Happens all the time, right?
Exactly.
Yeah.
If so, you've likely encountered an ingredient called erythritol. It's a really popular sugar substitute, chosen precisely because it has, minimal calories.
Mm-hmm.
But what if this ingredient, the one intended to support your health goals, could actually be having an impact on something as critical as the tiny blood vessels lining your brain?
Yeah.
That's the puzzle we're diving into today.
It's a fascinating area because, we interact with these substitutes daily, but understanding their precise biological effects, especially on something as delicate and, vital as the brain's microvasculature, that's still an ongoing process.
Absolutely. And the source material you've shared gives us a window into the latest piece of that puzzle. We have a health news article summarizing recent findings, and crucially, the actual scientific paper itself. This is a study from the University of Colorado Boulder, published in the Journal of Applied Physiology, and it's specifically titled "The Non-Nutritive Sweetener Erythritol Adversely Affects Brain Microvascular Endothelial Cell Function."
Wow, yeah. That title alone tells you pretty much exactly what they were aiming to investigate, how erythritol impacts the function of those specific cells.
Right. So our mission in this deep dive, really guided by the details in these sources, is to explore what this study actually did, what it found at a cellular level, what those findings might mean for you in the context of, broader health, and what questions it leaves for future research.
Getting past the headlines and into the actual science.
Exactly.
Okay, so maybe to start, let's quickly cover what the sources tell us about erythritol itself. What is it?
Sure. So it's described as a non-nutritive sugar substitute. It's a type of sugar alcohol, or polyol, with 4 carbon atoms.
Okay.
Its sweetness is about 60 to 80% that of regular table sugar, sucrose. And its key feature for dieters, really, is its very low calorie count, just 0.2 calories per gram. Super low.
And that low-calorie appeal is exactly why it's so prevalent today. You see it in things like protein bars, tons of low-calorie or zero-sugar beverages, and various foods aimed at managing blood sugar levels. The FDA approved its use back in 2001.
What's maybe less known, and the sources point this out, is that it isn't purely a synthetic compound. It's actually found naturally in small amounts in some fruits, vegetables, and fermented foods.
Right, like pears or grapes, I think.
Yeah, things like that. But perhaps even more surprisingly, it's also produced right inside our own bodies.
That's correct. Our body can actually create it from glucose and fructose through a metabolic route called the pentose phosphate pathway.
Huh.
So the amount of erythritol circulating in your blood is a result of both what you eat or drink and what your body naturally makes.
And that blend of, low calories and this sort of partially natural origin has made it a real go-to for people looking to cut sugar without completely sacrificing sweetness.
However, as the sources make very clear, some recent concerns about erythritol have emerged, and these were driven by earlier large-scale studies.
Yes. The news article and the scientific paper both reference prior epidemiological research. These are the studies that, observe health outcomes in large populations over time.
And some of the really prominent ones, like work led by Witkowski and colleagues, indicated a pretty concerning link. They found that people with higher concentrations of erythritol in their blood plasma seemed to have an increased risk of major cardiovascular and cerebrovascular events.
Events like heart attack and stroke.
Yeah.
Serious stuff.
Yeah.
The studies noted this association in different groups across the U.S. and Europe. And what made it particularly stand out was that the link seemed present even when researchers accounted for, the traditional risk factors for heart disease.
Right, like high blood pressure, cholesterol, etc. It seemed independent.
Yeah.
Furthermore, some of that prior work hinted at a potential mechanism, possibly related to blood clotting. Sources mention studies showing that soon after consuming a typical amount of erythritol, say, what might be in one artificially sweetened drink, maybe around 30 grams, there was an observed increase in the potential for blood clots and also greater reactivity in platelets. Platelets are the blood cells that help form clots.
So you drink this stuff, and shortly after, your blood might be, slightly more prone to clotting?
That's what some evidence suggested, yes. So there was this pattern emerging higher erythritol levels linked to increased risk of stroke and heart attack, and then some evidence of increased clotting potential right after consuming it. But exactly how erythritol might be causing these effects at a fundamental biological level, that remained unclear.
The why was missing.
Exactly. And that gap is precisely what this new study by Auburn R. Berry and colleagues at the University of Colorado Boulder aimed to address. They set out to investigate the cellular basis for these observations, focusing specifically on the brain.
Their goal, as stated in the sources, was to use laboratory experiments, working with isolated cells, what's called an in vitro study,
In the dish.
to look at the direct effects of erythritol on human cerebral microvascular endothelial cells. That's a mouthful.
It is, but they're incredibly important cells. They make up the inner lining of the smallest blood vessels in your brain. Their health is just absolutely crucial for maintaining proper blood flow, regulating what gets into the brain, and importantly, preventing issues like stroke.
Okay, so these specific brain blood vessel lining cells, what functions were they actually examining?
They focused on key markers related to blood vessel function and health. Specifically, they measured oxidative stress, the production of nitric oxide,
Okay.
the production of endothelin-1, and the release of something called tissue plasminogen activator.
And their hypothesis, I guess, based on those earlier population studies, was that erythritol would negatively impact these processes.
That's right. They predicted it would probably increase oxidative stress, reduce nitric oxide, boost endothelin-1, and maybe impair the release of tissue plasminogen activator.
Basically, they were looking for cellular signs that could explain why erythritol might be linked to increased vascular risk, correct?
Precisely. Does it mess with the cell functions known to be involved in stroke?
Okay, so how did they actually do this experiment in the lab?
Well, they took these human brain microvascular endothelial cells and grew them in culture. Then they exposed them to a single dose of erythritol.
And the sources point out the concentration they used was 6 millimolar. Why that specific number? Is that a lot?
It sounds technical, but the study authors state that this concentration is equivalent to the level you might find in someone's blood shortly after consuming that typical amount, around 30 grams, found in a single artificially sweetened beverage.
Ah, okay. So it's relevant to what someone might actually experience.
Exactly. So while it was a lab experiment, they were trying to use a concentration that's directly relevant to human consumption. The exposure itself lasted for 3 hours.
Just 3 hours.
Just 3 hours for this particular experiment, yes. After that, they analyzed the cells for changes in those key markers they were interested in.
All right, let's dive into what they actually discovered. What happened to these cells after 3 hours swimming in erythritol?
Okay. One of the really significant findings was regarding oxidative stress. Erythritol exposure caused a substantial increase in molecules called reactive oxygen species in these cells. About a 75% increase, actually.
Whoa. Okay, what are reactive oxygen species again?
Think of them as sort of unstable molecules, byproducts of metabolism. In small amounts, they're normal and used for cell signaling, but if you get too many of them, they can damage cells, lipids, proteins, DNA. That's oxidative stress.
And you said the cells lining the brain's tiny blood vessels are particularly sensitive to this.
Very sensitive, yes.
Yeah.
And the study found the significant rise, around 75% higher reactive oxygen species levels, showing the cells were definitely under increased oxidative stress after the erythritol exposure.
Did the cells try to fight back?
They did. The study notes they saw an increase in some antioxidant enzymes the cells produce, like superoxide, dismutase, and catalase, by maybe 25–45%. But the data suggests this wasn't enough to fully counteract the overall rise in those damaging reactive oxygen species. Increased oxidative stress in these specific brain cells is known to be harmful, potentially damaging the blood-brain barrier and just overall tissue health.
Okay, so increased cell stress. What about nitric oxide? You mentioned that before. We know that's crucial for blood vessel function.
Absolutely vital. Nitric oxide is key for helping blood vessels relax and widen vasodilation. That ensures healthy blood flow, and it also helps prevent clots and inflammation. Super important.
And what did the study find?
They found that erythritol caused a noticeable decrease, nearly 20%, in nitric oxide production by these brain blood vessel cells.
How did it do that? Did it just, destroy the nitric oxide?
Not exactly. The sources explain it's a bit more subtle. The total amount of the enzyme that makes nitric oxide, it's called endothelial nitric oxide synthase, or ENOS, wasn't changed.
Oh.
But erythritol seemed to interfere with its ability to work effectively. It basically altered the enzyme's activation state less activation, more inhibition in a way that significantly reduced how much nitric oxide it could actually produce. About a 33% reduction in the active form and a 39% increase in the inhibited form.
So the machinery was there, but someone threw a wrench in the works, basically.
That's a good analogy. And having reduced nitric oxide availability is a classic indicator of blood vessel dysfunction, what's called endothelial dysfunction, which is strongly linked to cerebrovascular problems, including stroke.
Okay, so we've got increased cellular stress and a reduced ability for blood vessels to relax.
Hmm.
What did they find regarding the third marker, endothelin-1?
Right, endothelin-1. It's essentially the functional opposite of nitric oxide when it comes to blood vessel tone. It's a powerful molecule that causes blood vessels to constrict or narrow, vasoconstriction.
So nitric oxide opens them up, endothelin-1 closes them down.
In simple terms, yes. Normally, they work in a delicate balance to regulate blood flow. But if you have too much endothelin-1 relative to nitric oxide, it can lead to unwanted narrowing and restricted blood flow.
And the study found erythritol increased its production.
Yes, they saw a marked increase, around 30%, in endothelin-1 production by the cells after the erythritol exposure. The sources point out they saw significantly more of the precursor protein for endothelin-1 being made, though the enzyme that converts it didn't change.
So the cell was gearing up to make more.
Seems like it. And the key takeaway here is this combination. Decreased nitric oxide, the relaxing signal, combined with increased endothelin-1, the constricting signal, that creates a profound imbalance that really pushes blood vessels towards constriction and impaired blood flow. And that's particularly problematic in the, delicate network of the brain's microvasculature.
Okay, that doesn't sound good. Finally, they looked at the cell's ability to dissolve blood clots using tissue plasminogen activator, or TPA.
Right, TPA. It's a crucial enzyme, really, that the body uses to break down fibrin, the main protein in blood clots. So it helps dissolve clots that might form in blood vessels. Very important for preventing blockages.
And what happened with TPA release?
The study looked at the release of this molecule and found something interesting. While the baseline level of release wasn't really changed by erythritol,
Okay.
the stimulated release was significantly blunted.
Stimulated release meaning like when the cell gets a signal that a clot might be forming and it needs to release TPA.
Precisely. They used thrombin, which mimics a clotting signal. In the untreated cells, this thrombin signal caused about a 25% increase in TPA release, deploying the clot buster.
Makes sense.
But the erythritol-treated cells, they showed almost no increase in TTA release in response to that same thrombin signal.
Wow. So their ability to actually activate their clot-busting response seemed impaired.
That's what the data suggests, yes. And having impaired ability to dissolve clots, which is known as impaired fibrinolytic capacity, is directly linked to a higher risk of stroke, especially ischemic strokes, those caused by blood clots blocking flow.
So, okay, let's put these 4 key findings together. The increased reactive oxygen species, the lower nitric oxide, the higher endothelin-1, and this impaired tissue plasminogen activator release. What do the study authors and the reporting sources say this all points to?
They conclude that these cellular changes, taken together, are well-established indicators, hallmarks really, of cerebral vascular endothelial dysfunction.
Dysfunction of those crucial brain blood vessel linings. We keep coming back to that.
Exactly. And the sources emphasize that this kind of endothelial dysfunction is considered a major underlying factor in the development, the severity, and the outcome of ischemic stroke.
So in essence, this study provides a potential biological explanation, a mechanism, for that concerning link seen in the earlier large population, studies the link between higher erythritol levels in the blood and an increased risk of heart attack and stroke.
It offers, as the paper puts it, novel mechanistic support for those prior epidemiological observations. They've basically shown in a lab setting that erythritol can negatively impact specific cellular pathways in brain blood vessel cells, and these pathways are known contributors to stroke risk.
And they also mentioned erythritol can get to these cells.
Yes, the authors also confirm that erythritol is known to be able to cross the blood-brain barrier, meaning it can physically reach these endothelial cells in the brain.
Okay, that gives us a pretty clear picture of what happened in the lab dish, but it's really critical, as both the study authors themselves and the news article highlight, to remember the limitations here.
Yes, these are vital caveats. Cannot stress this enough. This was an in vitro study done in a lab dish with isolated cells. It wasn't done in living, breathing humans or even animals.
Right, big difference. And the exposure was acute, meaning very short term, just that 3-hour window, using a single dose equivalent to consuming 1 beverage.
Precisely. The study, by its very design, cannot tell us about the effects of longer exposures, nor can it definitively speak to the impact of chronic, repeated consumption, which, let's face it, is how people typically consume these products day after day, maybe over years.
So while the findings are definitely significant because they point to a potential mechanism, a possible why, we absolutely cannot draw definitive conclusions about the clinical risk for humans solely from this one study.
That is absolutely the correct interpretation. This study provides crucial data at the cellular level. It aligns with and helps explain those prior population findings, which is important, but it does not, on its own, establish cause and effect in humans or quantify long-term risk. It needs more research.
And that leads us right to the call for future research mentioned in the sources. What needs to happen next to truly understand the implications for human health?
Well, both the scientific paper and the news article strongly advocate for more research using long-term studies and also in vivo models, that is, studies conducted in living organisms like animals. And critically, they emphasize the urgent need for well-designed clinical studies in humans to clarify the potential consequences of habitual dietary exposure to erythritol over extended periods.
Because the impact on someone who has, say, a diet soda with erythritol every day for 10 years might be quite different from this single 3-hour exposure in a lab.
Exactly. We just don't know yet. This study provides compelling evidence that there's a cellular pathway worth investigating much further, and it gives researchers specific targets like nitric oxide function and TPA release to look at in those future, more complex studies.
So let's bring this back to you, the listener. This deep dive has taken you right inside the complex world of your brain's tiniest blood vessels. We've seen how, based on this recent lab research, a common ingredient like erythritol, at a concentration achievable after drinking just one diet beverage, negatively impacted key cellular functions. And these functions are known to be linked to increased stroke risk.
It really just underscores the intricate connection between the ingredients we consume, even ones marketed as healthy alternatives, and the microscopic processes happening constantly within our bodies.
So while the scientific story isn't fully written yet, clearly more long-term human studies are needed. Given how widely used erythritol is, often in products chosen specifically for health reasons.
Yeah.
What does this research prompt you to consider about the ingredients in the foods and drinks you consume regularly, especially, as the science continues to evolve?
Yeah, it's definitely food for thought while we wait for more data.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.